Okay, I'll make a couple of comments while we're waiting. So the topic suggested for me this morning, I've expanded a little because it was specifically on building family communities to enhance knowledge and improve patient care. I have been interested in the topic of data sharing to enhance knowledge and speed knowledge and improve patient care. So I'm going to broaden the topic to talk about engagement and interaction with clinicians, clinical laboratories, and families, because many of you have more experience interacting with families and engaging them than I do. So I will get to that original topic, but start with the role and responsibility of clinicians and clinical laboratories. I should mention I am a consultant and member of Scientific Advisory Board for the companies listed here, although I won't be talking about any specific products or services today. So I got interested about 5 years ago when chromosome microarray whole genome analysis became available. And it was clear that we'd be doing tens to hundreds of thousands of whole genome analyses on patients as a clinical fee-for-service test. And that meant that generating whole genome data was essentially free as a byproduct of patient care if we could just capture that data in a useful form. Meant that there was more structural variation or CNV data generated as a byproduct of patient care than in all the NIH and other funded research studies in the world, and so we just had to catch that free data and store it someplace that was in a useful database. And the same is rapidly becoming true for exome and whole genome sequencing. There will be way more sequence data generated as a byproduct of patient care within the next couple of years. So we just need to create the infrastructure and process to get that data into a database for research data mining to rapidly increase our knowledge and apply that to improved patient care. So how do we capture and leverage that? And the reason that this is important is with these new technologies, looking at the whole genome, as you're all aware, we don't always know what a structural variant or a sequence variant means, and we want to know whether it's clinically relevant, pathogenic, or it's benign and a normal polymorphism or no phenotypic effect. And you're familiar with the way we go about it. We hope that there are examples in the literature that tell us whether it's a known disease, known genotype-phenotype correlation or not. And if it's not, then we use our clinical judgment based on the number of genes or the particular genes and family information about de novo versus inherited status. But if we make clinical judgment decisions, that allows for a lot of variability from individual clinical laboratory director, or clinician. And that means there can be error rates due to the subjective nature. So we really would like a whole lot more data on genotype-phenotype correlations and an evidence-based review process to establish which are the pathogenic variants and which are the benign variants and get rid of this much larger bucket in the middle of variants of uncertain, unknown clinical significance. So 5 years ago, Krista Martin and I at Emory started a initially small collaboration between a couple of labs that quickly became an international collaboration between many cytogenetics lab, a voluntary community effort to collect genotype and phenotype data in a standard way and to submit it to a central repository at NIH in Bethesda and make it available to all researchers and all clinicians. These are the laboratories now, over 190 worldwide, that are members and contributing data. This, in fact, pretty rapidly proved itself to be very valuable. The first data analysis published last September by Aaron Kaminsky and Krista's group at Emory on the first almost 16,000 cases compared to normal controls allowed us to classify 21 pathogenic CNVs. One example is a duplication of 16p11.2 that when we started the analysis was a variant of uncertain clinical significance. But by comparing large data sets to normal controls, we're able to statistically, in a statistically significant way, show that this was a pathogenic duplication. Similar studies by Evan Eichler's group on data from signature genomics was another 15,000 has contributed to this, and in total there are over 30,000 clinical cases that have allowed us now to update the known pathogenic CNVs by Array CGH and other platforms. So on our website for the ISCA consortium, there's information about how to submit data. The goal of this is to make it easy on the clinical labs, and the notion is a one-click solution, meaning at the end of every day or the end of every week, you push a button and says submit all of our patient data to this public free database. We're not quite there yet, but making good progress. And the way that we're doing that is working with all of the major vendors who provide the hardware and software for clinical laboratories to do whole genome microarray analysis, and asking them to provide the tools to their customers for this one-click data submission so the busy lab can accomplish this without interfering with their workflow, and then they don't have any excuse for not participating in this important project. Now, these are slides I borrowed from Krista, because one of the benefits of this data sharing project that I guess if somebody had asked me ahead of time, I would have said, yes, it's probably going to help quality control in the laboratory, but I didn't fully appreciate the magnitude and importance of this. And what this means is NCBI has written some data quality checks, which means one laboratory submitting data. If you submit a CNV one day and then 3 months later submit the same CNV and you classify it differently, so one time you call it pathogenic, the next time you call it unknown, it'll automatically flag that and send you an alert back to say you've sent the same CNV twice and you classified it differently. Would you like to reconsider the classification of one or the other to reconcile this discrepancy? The frequency of those discrepancies within our lab at Emory blew me away, because we had all talked about our criteria for classifying. We all sat in the same analysis room, shared experience on a daily basis, and yet we still had different classifications from a single lab. Then when you compare the data between laboratories, and so now we have NCBI has our known pathogenic track available to them. So if you send in a call and you've classified it differently than the known pathogenic curated call, it'll flag that, send you an alert and say, this is in the category of known pathogenic, you've called it of unknown significance or even benign. It'll give you an alert and give you an opportunity to change your mind or comment why you classified it differently. The early analysis of this shows that this is about 5% of calls, or there's a 5% discrepancy rate. This is huge and this is scary. So this benefit immediately to any lab submitting its data to find internal and across laboratory discrepancies as a quality control measure is huge, and it's much more important than I anticipated. So this now I'll add quality control to the normal list of data sharing motivations and incentives. So how do we get patients, clinicians, laboratories to share data with each other to enhance knowledge? So there's the usual altruism category that gets us some distance. There's co-authorship or co-funding related to grants that can help. We submitted one paper on the ISCA project, and one of the reviewers said, well, given the newness of the technology, the importance of the problem, it would be unethical for a clinician not to provide clinical phenotypic information when ordering this test to allow us to improve our knowledge and improve patient care. And it would be unethical for laboratories not to share their data. Quoting the reviewer here, but I tend to agree with that reviewer. But the quality control factor I now think is by far and away the most important. And I think it's so important that I'm starting to advise my colleagues who are ordering this test that you should not only pay attention To the lab, you're sending your genetic testing to a CAP approved, CLIA approved, but if they're not participating in a quality control program, QA/QC program, like the ISCA consortium, and not benefiting from the error checking and quality control, that may not be the best laboratory to submit your samples to. And this is part of the paradigm for data curation resulting in evidence-based review and practice guidelines that just says any individual lab by itself, regardless of how big and how many cases and how many experts, is just a single source of curation and prone to some discrepancy, bias, etc. Comparing your data with multiple laboratories improves it, but then to have a panel of experts to review that data and then to develop these into practice guidelines or professional societies sort of blessed known pathogenic, known benign variants. So this whole genome microarray analysis, I think, has been a very successful community project to share data, rapidly increase our knowledge, and therefore improve the quality of chromosome microarray clinical testing. And as a model, I think it's been so successful, it will now inform the much larger problem we're going to have moving into whole exome and whole genome sequencing. So our ISCA consortium has actually approached the molecular diagnostic DNA sequencing community, proposed a partnership. We've written a large grant together that's pending review, and we're optimistic about potential funding early next year to create a combined clinical genomic variation database that will have structural and sequence data along with phenotypic data as a research data mining resource, but more importantly to improve clinical interpretation of sequencing and CNV data. This is being led by Heidi Rehm, the head of molecular diagnostics at Harvard, and involves a number of molecular diagnostic sequencing labs in the United States. The Genome Institute, which would be the funder of that database, has also recently published a request for application for a major NIH grant on clinically relevant genetic variants resource. And this is going to be one grant. It's about a $14 million grant over 5 years to support the developing evidence-based process and expert curation to identify which variants, sequence and structural, are ready for prime time and clinical application from pharmacogenomics, Mendelian genetic disease, and common genetic disease association. This RFA just came out, and it's interesting, there's a requirement in the RFA to partner with the American College of Medical Genetics, so Mike Watson is busy working in discussions with people about how the college will participate in this, and there will be a requirement for many medical genetics and molecular genetics diagnostic lab experts to participate in this very large and important project. And we were pleased to see that in the RFA they said one of the existing models for this kind of data collection and data curation is the ISCA project, and that whoever wins this award should partner with and collaborate with the cytogenetics CNV project already. Now I'll switch a little bit to engaging patients and families. From this whole genome microarray data, there have been a number of new CNV disorders or microdeletion disorders that have quickly come to attention. We can actually list them in terms of frequency. So 22q11.2, a well-known microdeletion, is still the most common. The second most common is this newer deletion of 16p11.2, initially reported in association with autism and development disabilities. This got the attention of the Simons Foundation, the largest private funder of autism research in the world, and they set up something called the VIP project for variation in individuals. The notion of this is to break the heterogeneous group of disorders that comprise autism into genetic subtypes, starting with this one copy number variant. And one of the first things they did was to set up a website and an association, in part for the benefit of families, so that families with this deletion would not feel isolated and alone, but would immediately have access and ways to contact and communicate with other families. In fact, the second annual family conference took place last weekend in Chicago and had over 100 families there, I think. There's a family matching service to get families with similar age children and other issues together, and allow them access, if they're interested in clinical information and research information, and also to allow researchers to get access to a number of families who have agreed to be contacted and participate in research. This is the graph of the recruitment of families just in the last year or so for this project, the map of the sites of the families recruited into the project, age distributions, and one of the nice things about whole genome chromosome microarray is we're able to identify a significant number of very young children, which we hope eventually will allow us opportunities for early intervention and prevention of some of the symptoms, in this case, features associated with autism. And recent numbers for this project: 433 total registrants, 200 patients with deletion 16p. So this is showing us that, unlike in the past where it took many, many years to identify enough families to do statistically significant research on phenotype and natural history, et cetera, that with some resources, but also by engaging families and particularly with availability of the internet, you can more rapidly identify a larger number of families and start accumulating information for research. These are some of the features on the website that I'm not going to talk through, and that, as I said, there are ways for families to connect with each other by geographic location, age of the child, etc. And then they use this online registry to conduct surveys of families, and you can view the data. This is similar to an earlier project that Andy Fawcett, who was at Emory and now at Geisinger with me, a senior genetic counselor, worked with the Parent Project Muscular Dystrophy Organization to start Duchenne Connect, which is now an international registry involving 37 countries in partnership with TREAT in MD, a parent organization for muscular dystrophies. Some of the information, and now for Duchenne and some of these other patient registries, they now become a resource for pharmaceutical companies and researchers to recruit for clinical trials and make available to families the possibility of pharmacologic trials and treatment. The Office of Rare Disease Research at NIH earlier this year had a request for applications to create a global rare disease registry. This RFA was responded to and awarded to a small informatics company called Patient Crossroads. Many of you are familiar with Kyle Brown, Vanessa Miller, and through their early work with Duchenne Connect and also working with Ronnie Singh and SERGG to develop NBS Connect for newborn screening disorders, they've now been awarded a grant to develop a more global rare disease registry. And these are all of the reasons for the interest by NIH as well as for us in patient registries. And as Kyle says, thank you, I'll wrap up. All patient family support groups are interested in partnering with industry, including pharmaceutical company, but they're handicapped when they go to these pharmaceutical and other companies and say, would you like to partner with us and help us? And if they can't even answer the simple questions, how many patients and families are affected with this condition and where are they? So you've got to build a registry to know how many families, how big is the problem, how many families are there, and how can you get in touch with them to discuss with them participation in research and clinical trials. And everybody's goal here is to improve diagnosis and improve patient care and outcomes, as well as providing the other kinds of social support I alluded to earlier. So this is the model for the Global Disease Registry, which will interface with individual disease-specific registries and accumulate de-identified data in a single place that's accessible by researchers to do much more global kinds of research. This is analogous to another effort patients like me, many of you are familiar with, and I just put this slide in to say the key for all of this is more data, and I'll just conclude with the responsibility, obligation of sharing data and sharing patient data is important to improve patient outcomes by putting patients first. That's not just the mission of patients like me, it's all of our mission. So I'll stop there just by acknowledging my longtime collaborator, Krista Martin, and her group at Emory that have been involved in all of the components of my talk today, and Andy Fawcett, who I stole from Emory and moved him to Central Pennsylvania to continue this work in close collaboration with Emory. So I'll stop there and answer questions. Thank you.
Any questions?